Hi, good morning, everyone. Welcome to our webinar, Hammering Out the Single-Cell DNA Sequencing Solutions to Advance Solid Tumor Research. I'm Joyce Chen, Channel Marketing Manager at Mission Bio. I will be your moderator today. Here are just a couple of reminders before getting started. We are currently recording the session. Also, at any time of the webinar, you can type in your questions in the chat box and we will ask the speakers at the Q&A sessions after their presentations. So it's my pleasure to introduce our speakers, Dr. Ania Wonsky, who is the Director of Market Development Applications and Collaborations team here at Mission Bio, and Dr. Divya Wao, who is the Senior Manager, again from the Market Development Applications and Collaboration team at Mission Bio. They are going to share with you how to utilize single-cell DNA sequencing for solid tumor research. After that, Dr. Lei Tong, who is the Staff Regional Marketing Manager at Illumina APJ, and she is going to talk about how to unravel cancer's complexity on Illumina platforms. All right, with that, I would like to hand it over to Ani and Divya.
Thank you so much, Joyce, and welcome and thank you for joining us for this webinar. So I'll give a quick introduction into Mission Bio and then I will give it over to Divya, who will then go into more detail about some of the ways that you can use single-cell DNA sequencing for solid tumor research with some pretty amazing data and case studies from both our own work as well as our customers. So at Mission Bio, our mission is to unlock single-cell biology to enable the discovery, development, and delivery of precision medicine. And what does that mean? So we're trying to make single-cell DNA sequencing and multiomics, because we can do proteomic work, but we're not going to be talking about that from a solid tumor perspective, mostly in oncology and in cell and gene therapy. But this is really just the beginning. So watch this space as we have lots of exciting developments that are also coming down the road. So let's talk a little bit about who Mission Bio is. So we are based in the San Francisco Bay Area, just north of the airport. We've sequenced over, we've helped our customers and ourselves, we've sequenced over 10 million cancer genomes. We have over 34 custom panels and over 800 users on our software, our Tapestri platform or portal. And in the United States, over 50% of the national cancer institutes, these are a national funded institute by the National Institutes of Health, have adopted the Tapestri system for their work. We have a number of distributors both in the Asia Pacific area that can help you in local languages as well. So if you do have any questions or would like more information, please reach out to us and we're happy to either help you or connect you to one of our partners that is able to assist you both in your local time as well as in your local language. So why do we need single-cell DNA sequencing and why specifically single-cell? Well, we all know that cancer is a very complicated disease and it is a progression from normal to malignancy with these genetic changes that occur over time. And what we understand from decades of research is that these changes accumulate over time in these populations. But what we aren't able to tease out particularly well with traditional, what we call bulk next generation sequencing methods, is how these changes are accumulating and what that progression and lineage really looks like. And so to be able to understand the disease more informatively in a much more comprehensive way, we need to be able to look at these cells at a single cell level at their genomic profiles. And we'll give you some examples of this in this talk as to how we're able to see that and the kind of definition and insights that we're able to identify from these cells when we're able to get to this level of granularity. So why can't we just do bulk sequencing? So bulk sequencing is an average of what the population holds. So for example, if we take this population of cells that has a number of mutations, each of these circles are a cell, and some have a single mutation and some have double mutations. And so if we did bulk sequencing, the readout here would be that 40% of the cells carry one mutation, and then 40% of the cells carry the other. We don't get the clarity that we can with single cell analysis that tells us how many and which proportion of those cells carry only one mutation versus the second mutation versus both mutations. And this is obviously very important when we're trying to identify treatment regimens, which populations may cause recurrence or resistance mechanisms, as you'll see in some of the examples that we'll show. And we know this is incredibly important because even if we are very, very good at treating disease, a lot of the time that relapse that we get in those patients is driven by those resistant clones. So they're clones that existed at the very beginning. They may be rare populations only present, and we have examples of this where there was only one cell present that ended up becoming 70–80% of the tumor at the remission stage. And so we really want to be able to understand what's happening from a biological perspective, so we're able to treat these populations as early as possible and prevent that relapse from occurring in the first place. And so single cell DNA analysis is really the only way that we're able to get down to that level of detail so that we're able to identify those particular mutations and then plan for that from a treatment perspective. So how do we do single cell sequencing? So bulk NGS, you have one sample, you just read all of those samples and you measure it. With the Tapestri system, we can barcode each cell independently, pull those back together, sequence them by Illumina sequencing, and then we're able to measure the co-occurrence of those mutations with every cell and bioinformatically determine which cell had which mutation. So why is this important? So for example, with bulk NGS, you're really inferring whether or not a mutation exists within that population versus actually quantifying on a single cell level, because we're able to actually assess whether or not that mutation exists versus giving a percentage of that population, because we're doing it at a single cell level. With zygosity, we're able to also quantify this because we're able to get down to that resolution, and as well as clonal architecture, obviously mutational co-occurrence, which we're talking about, and whether or not we have cells that are not cancer cells. We're able to take that out of the population mix, and that is also a pretty big part, especially in solid tissue where you may have a lot of stromal cells. We're able to be able to pick those out bioinformatically, and so they don't contaminate your sequencing and take up all of those runs. Again, like we mentioned, we have a lot of capabilities from a resolution perspective. Bulk NGS, you only get that one mutational profile, which is that average of the whole bulk population, versus single cell, which you're able to resolve up to 10,000 different mutational profiles, up to however many clones that you're able to capture. And we can look at single nucleotide variants, insertions, deletions, copy number variants, as well as loss of heterozygosity. And then for if you're doing a bit of heme work, as well as some solid tumor, we're able to look at protein expression as well, extracellular protein expression. And so what can we use Tapestri for? So like I mentioned, it's primarily being used for clonal mosaicism and looking at what happens from a transition perspective upon malignant transformation. We can look again at that clonal evolution in primary tumors as well as in xenograft samples, and Divya will go into some of the sample types that we've had customers be able to run. Obviously, we can look at things like metastasis, epithelial to mesenchymal transition or EMT, as well as whether or not we're getting drug resistance and what those mechanisms are. And so we're able to do this by measuring the genomic progression of those cells, like I mentioned, whether it's single nucleotide variants, insertions, deletions, changes in copy number, loss of heterozygosity, and again, we're able to see that mutational co-occurrence, which is really, really important from a therapeutic perspective, and we're able to get that rare cell population of down to 1 in 1,000 cells within that population. So with that, I'll pass it on to Divya, who will give us a little bit more insight into the types of work that people are using Tapestri for solid tumor.
Thanks, Anya. So single-cell DNA sequencing using our Tapestri platform enables researchers in the heme, solid tumor, and cell and gene therapy space to unambiguously identify variant zygosity, uncover patterns of mutational co-occurrence, and detect rare mutations in thousands of individual cells. Now, with this in-depth insights, researchers can determine how cells transform from benign to malignant states, like Anya pointed out, reveal genomic changes enabling cancer cells to metastasize, and elucidate the mechanisms of drug resistance. So meet the Tapestri. This is a novel multi-parametric platform that enables you to analyze SNVs and indels. And now we have the multi-omic approach to be able to look at CNVs and surface protein within a single workflow that both conserves sample and consolidates multi-omic assays into an integrated single cell resolution data set with easy to use analysis software. The Tapestri seamlessly wraps around standard Illumina NGS workflows to enable novel single-cell multi-omic resolution. In addition to our easy-to-use instrumentation and fully kitted reagents, we provide a suite of software to support upfront custom DNA panel designs, as well as an integrated Tapestri pipeline for multi-omic analysis, and finally, Tapestri Insights for data visualization and publication-ready figures. So let's look at how our technology works. Now, this is based on a novel two-step microfluidic workflow. So individual cells or nuclei, they undergo encapsulation to form isolated droplets that contain protease enzymes to allow access to genomic DNA. The lysate, including the now histone-free DNA, undergoes a second encapsulation step where the contents are merged with barcoding beads, PCR primers, and reagents to target specific regions of the genome. Now, sequencing libraries are then prepared and sequenced on any of the Illumina platforms. As Anya mentioned, we're capable of doing surface protein. So for surface protein detection, those antibodies targeting the surface protein are conjugated to oligos. There is a brief antibody staining protocol up front of the cell encapsulation step, and then the antibodies are subsequently digested during the first encapsulation protocol, leaving behind just that oligo barcode within the droplet. And this oligo is then captured and integrated into the genotyping library and reported out as a corresponding NGS read after sequencing. And as Anya mentioned, there's different sample types that we support as on the previous slide, Anya. There's a fresh frozen OCT embedded. We can support cell lines, mouse samples, clinical samples from both solid and heme malignancies. So we have several panels to fit your application needs. Our catalog panels are pre-designed and wet lab tested to get your lab up and running with minimal time and effort. Our published panels are designed by leading researchers and verified for performance through a peer review publication. Now, if you require more flexibility, you can consider the pre-designed and customizable virtual panels. Custom panels can be created for human, mouse, or any other reference genome of your choice. So moving on to customer data here, the Tapestri platform is empowering cancer researchers in the solid tumor space to ask and explore questions that were previously inconceivable. This is a recent paper in Nature from the lab of Pirolito from Memorial Sloan Kettering Cancer Center, where he sought to identify the genetic basis of resistance to sotorasib, a drug that was approved for treatment of patients with advanced non-small cell lung cancer. These patients carried a KRAS G12C mutation. Now, despite the encouraging clinical results, a vast majority of patients did not respond to this inhibitor therapy, mainly due to an acquired genetic resistant mechanism. So this group wanted to understand what were the genetic mechanisms for the resistance to this targeted therapy. Now, although bulk sequencing identified a host of secondary mutations that arose during treatment in several genes of the RAS signaling pathway, like you can see here, the KRAS, NRAS, MRAS, and BRAF, it lacked the ability to uncover the co-mutational relationships within cells. Resolving co-mutational patterns is fundamental to understanding how the secondary mutations bypass this targeted therapy. As our single-cell DNA sequencing technology is the only method that can definitely measure the co-occurrence of mutations within cells. And using this platform, the researchers found that the secondary mutations they identified from bulk sequencing often co-occurred with the KRAS G12C mutation in the drug-resistant cells. Now, these secondary mutations within the same cells were shown to circumvent the effect of the targeted therapy by upregulating other players in the pathway. So the authors go on to conclude that resistance to sotorasib can be delayed by co-targeting intermediates in the MAPK-ARK pathway, a finding that may have clinical significance. We now have several leaders in the solid tumor space that have leveraged our platform to understand the genetic heterogeneity underpinning cancer, uncovering rare cell populations, co-mutational patterns, and zygosity that drive the onset and progression of these cancers, and understanding the genetic mechanisms of resistance to targeted therapy that can lead to relapse, such as this particular paper. So let's move on. I'd now like to present some data that we validated on clinical breast tissue samples to test our workflow and see if we could detect biologically relevant genetic changes in clinical tissue. The samples we procured were all infiltrating ductal carcinomas and were all grade 2. And we used our single nuclei suspensions to generate nuclei from these tissues. This is a newly optimized protocol. To these samples, we spike in nuclei from Raji cells, which is required for the pipeline for CNV baseline calling. And lastly, we leveraged our close relationship with our first solid tumor center of excellence, Dr. George Wies-Feho at Memorial Sloan Kettering Cancer Center, to design a highly biologically relevant panel specific for breast cancer. So let's take a look at the panel. This panel targets about 32 hotspot genes and about 30 copy number variants, both at the gene level and the chromosomal arm level. The panel was informed by the MSK impact data set and more than 15 databases, such as the TCGA and COSMIC. And it is designed to cover some of the most commonly mutated genes associated with breast cancer, including BRCA1 and 2. Moving on to sample prep here. The single nuclei suspensions were extracted according to our optimized enzymatic dissociation protocol in our user guide. Just briefly, sections of the tumors, they get minced using a scalpel and then incubated with lysis buffer. The lysis solution is then inactivated and the resulting slurry is strained and centrifuged to capture nuclei. The pellet is then resuspended in a solution, a spermine solution, to minimize nuclei aggregation, filtered, centrifuged, and the nuclei counts were obtained from DAPI. In order to cover or analyze the copy number variation, the pipeline first calls clones on the basis of SNVs. The pipeline maps the sequence reads to the genome, assigns the reads to cell barcodes, and performs genotype calling. And then the clones are identified using SNV data as seen in the heat map here. This particular patient had one clone, and we can see a second population of our spike in Raji cells as well that were also identified. To call ploidy, the reads are normalized to the spike in Raji diploid genome. And now we're looking at the chromosomal changes at the arm level. Each dot here represents an amplicon assayed using our single-cell DNA platform, and the panel is designed so each target has a minimum of 5 amplicons, and at least 5 of them must show a relevant change, that is either a copy gain or a loss, for the pipeline to accurately call CNV changes. Now, in this particular subject, there was a gain of chromosome 1q, 7q, chromosome 8p, and a loss at chromosome 13q. And at the gene level, this patient had a gain in EGFR and a copy loss in BRCA2 and RB1 genes. Now, EGFR is implicated in breast cancer progression and is associated with an aggressive phenotype, while BRCA2 and RB1 loss has been associated with poor prognosis. So as you can see from our internal data here, the Tapestri was able to simultaneously call some biologically relevant SNVs and gene as well as chromosome arm level copy number variations in clinical tissue at the single cell level across 10,000 cells. I'll now hand it over back to Anya.
Thanks, Divya. So as Divya just very nicely showed, we're able to use Tapestri in multiple different ways in solid tumor research. And obviously some of the examples that we've used are in breast cancer and in lung cancer, but you're able to see how we're able to identify those drug resistance mechanisms and really be able to tease out the genetic complexity of what's going on in these samples. So please, I see the questions coming in, so please keep them coming. We have a full solution for solid tumor research, and obviously breast cancer is a big focus as well. We have that optimized nuclear prep protocol, which was just released at the end of last year. That panel that we used for, that was in collaboration with Dr. George Rees-Philo from Memorial Sloan Kettering, is now one of our catalog research panels. So if you're interested in running that, that is available today. We also have the tumor hotspot panel. And as Divya mentioned, you can customize the panels as well. And we do have a partnership with another company called S2 Genomics to help enable sample prep. So if you do have trickier samples that you're trying to run, we can certainly help with that as well. Obviously, the instrument that all of this is run is called the Tapestri, and then we have an analysis pipeline, both to do the primary analysis, as the secondary analysis, as well as the tertiary analysis for the copy number variation, and that is done through our field application scientist team. So with that, we will answer any questions that is in the chat. So thank you for putting those in. If you do have any others, please feel free to throw them in as I hand it back to Joyce for the Illumina presentation.
Yeah, thank you, Anya and Divya. So the next one is Dr. Tong from Illumina APJ.
Thank you very much, Joyce. Let me try to share my screen. Sorry, give me a second. Can you see my screen?
Yes, please go ahead.
Okay, I'll put it in full screen. Okay, thank you all for being here. My name is Lei, and I'm the oncology marketing manager for Illumina Asia Pacific region. And I would like to thank Mission Bio first for inviting me to the great event. And after the two fantastic speakers sharing the exciting single cell analysis solutions from Mission Bio, I will be expanding the topic slightly with a brief overview of the multi-omics approaches in cancer research using next-generation sequencing technologies from Illumina or NGS, and how those technologies have accelerated our understanding of cancer, and also how NGS has changed substantially over the last few decades to become this incredible, flexible platform that can help us thoroughly understand any level of cellular regulation and also characterize the inherent complexity and heterogeneity of tumors. I would like to give a brief intro of Illumina to those of you that may not know us. So we are a company that develop, manufacture, and market innovative next generation sequencing and microarray technologies. Our goal is to enable the researchers to make the foundational discoveries, transform medicine, and ultimately impact human lives. Today, there are more than 220,000 publications that are featuring the Illumina NGS technology. So you can access our technology in two different ways. You can either outsource to a regional service provider or by implement the in-house sequencer from Illumina. So before we jump into NGS and multi-omics, let's first answer the question, what are omics? And as you likely know, the central dogma of biology details how information encoded in an organism's DNA eventually becomes a phenotype. And crucial information is encoded in the cell's genes, then transcribed into RNA, which is then translated into protein. And of course, regulation of how DNA is transcribed is often controlled by the non-genetic regulatory mechanisms, such as epigenomic regulation. So each layer of the central dogma, the genome, epigenome, transcriptome, and the proteome are considered as oms. And omics is an umbrella term referring to the study of individual or multiple levels of the central dogma. The current and growing NGS solutions can generate massive quantity of information about any omic, alone or in a combination. So starting at the genome, we know that cancer and mutations are fundamentally linked, and that profiling mutational landscapes is a phenomenal way to assess the susceptibility to cancer, predict prognosis, classify tumors, or diagnose the disease. So whole exome sequencing remains as a great approach to assess the genes that are most frequently aberrant in cancer, because those are the genes that are transcribed into mRNAs. But it's becoming clearer that 99% of the human genome that is non-coding also has associations with cancer. So whole genome sequencing is becoming increasingly prevalent in cancer research. Alternatively, even novel analytes like circulating DNAs released into the bloodstream from tumors can now be analyzed on NGS devices. A second increasingly popular application of NGS is epigenomic profiling, which can provide mechanistic context, for example, DNA methylation, for how the genome is regulated in cancer. So understanding how the genome is organized and regulated epigenomically is often critical for understanding how cancer is regulated. And that can now be accomplished using NGS via applications like ATAC-seq or METHO-seq. And RNA sequencing remains the most common applications of NGS because transcriptomics connects the genome to gene function, and measuring RNA most frequently provides a functional context to our understanding of samples. And importantly, detection of altered pathways may identify therapeutically exploitable mechanisms. RNA sequencing via NGS has also expanded to include analytes like non-coding RNAs, microRNAs, or circulating cell-free RNA transcripts. And finally, one of the most exciting emerging applications of NGS is proteomics. Proteins link genotype to phenotype by directly measuring the functional consequences of genomic, epigenomic, and transcriptomic regulation, as we mentioned earlier. And in cancer research, proteins have been crucial as biomarkers to specific cancers. So identifying cell membrane protein biomarkers could tell us whether a tumor or cancer cell will be responsive to immune regulation or immune therapy. And there are a number of methods that enable proteomic analysis with NGS at unparalleled complexity and sensitivity and scale. And any of those omics can now be analyzed on one NGS platform using the infrastructure and instruments likely already present at your institution or the regional service provider. But NGS flexibility and potential uses for cancer research aren't even limited to intracellular regulation. As Anya and Divya mentioned, right? So tumor cell populations are also incredibly diverse. and don't consist of one cell type. Rather, tumors are a complex ecosystem of different cell populations that evolve, change over time, and interact with non-cancerous cells, such as immune cells or stromal cells. And this cellular diversity, also known as a cancer microenvironment, really matters for regulation of cancer. And traditionally, sections of tissues or a population of cells are sequenced together in a method called bulk sequencing because many cells are lysed at once and sequenced at once. So this approach reveals the average gene expression among the population of cells. This remains a relatively simple, cost and time effective and a reliable way to profile the cancer samples. However, since the cells within the same cancer sample are heterogeneous, so next generation sequencing at the resolution of a single cell adds increased resolution to analysis, which has been perfectly highlighted by the two speakers before me. And if even greater resolution is required to understand the cancer microenvironment, there is an emerging method called spatial sequencing, which enables thorough characterization of cells within the context of their location within the tissue. So, for example, your goal was to determine whether immune cells are infiltrating a tumor. Single cell sequencing may not necessarily reveal all the information, whether immune cells are in the right place for immunotherapy efficacy, but spatial can provide that additional context. So we have seen this incredible transformation of next generation sequencing technology. The unique capability of NGS applies unbiased discovery power to any level of cancer regulation and at multiple levels of resolution, positions cancer research for major future breakthroughs, both in understanding the fundamental aspects of biology and in enabling future therapies. So NGS has its origin in DNA and RNA-seq and has enabled comprehensive characterization of unique cancers because of its unparalleled capacity to remove the bias from research and capture characteristics that were previously unforeseen or even unexpected. And the goal of all this multi-omics information is to move cancer treatment toward a future where not only can we have treatment specific to different cancers, but also to understand each patient's disease in such a detail that can be stratified and given personalized therapies for their particular disease tailored to their omics information. And that is why it's unsurprising that cancer research has become the most common application of multi-omics, and that multi-omics has experienced tremendous year-over-year growth. We are currently seeing a 63% year-over-year increase in publications, including multi-omics studies. We are also seeing a 48% year-over-year increase in grants for multi-omics studies. And this is reflective of research across all fields, suggesting that combining the characterization of multiple levels of cellular regulation enable researchers to observe things that wouldn't be hidden with only one type of data. And so with the NGS technology, you can unlock a wide array of applications and a wide menu of insights. And shown here on the slide are the main representatives of the Illumina sequencing platforms. From the left side, we have the MySeq platform, which yields 15 GB of data per run and is ideal for targeted sequencing, small genomes, or metagenomics. And next to it is the NextSeq 550, which yields about 120 GB of data per sequencing run, and it can accommodate RNA-seq, low throughput single cell analysis, some enrichment studies, etc. And I would like to highlight the next instrument of NextSeq 2000, which is the newest member to our desktop sequencing family. So per run, it can yield about 300 GB of data, and it's an ideal platform for in-house implementation that can cope with most of the applications, the multi-omics approaches that we discussed today. such as RNA-seq, single-cell-seq, proteomics, spatial sequencing. And last but not least, we have the NovaSeq 6000, which with its max sequencing output at 6 TB, and is ideal for high throughput or population scale of studies. And you see this type of sequencer most common at service provider labs. And is ideal for high throughput single cell studies, high throughput RNA-seq, metagenomics, epigenomics, and high throughput proteomics, etc. So as we see the throughput increase across the sequencers from left to right, it will enable you to do more samples, newer applications, and at a greater sequencing depth, either per sample or per cell. And no matter what you are interested in running, which modality, and whether you want to do a global study or select your targets of interest or adding the spatial context, and no matter the scale, whether you are studying hundreds of cells or millions of cells, we will have a sequencing solution that fits your needs and are more than happy to assist you in selecting the optimal solution for your project or for your lab. With the limit of time today, we cannot really deep dive into the workflows or some of the recent examples of findings that are enabled with the multi-omic method that we briefly went through today. I would like to provide this really great resource of a multi-omics method guide. If you scan the QR code, you'll be able to find this resource with a collection of end-to-end workflows for the most popular multi-omics. It conveys information about the multi-omics, provides some of the most recent multi-omics research articles, and shows the end-to-end workflows for the most common omics combinations. And with that, I would like to thank Mission Bio again for inviting us to participate in the event, and thank you all for your attention. Feel free to leave me any questions in the chat window.
Great, thank you, Dr. Tong. All right, we would love to take any questions you have. I see some questions coming in, Anya and Divya is answering them. You can continue to submit your questions to the chat box, or you can also unmute yourself and talk to us. Any questions, suggestions, thoughts need to come in from our audience? Hey, Eric. I just unmute you. You could please feel free to speak out and raise your questions. Hey, Eric.
I just wanted to, as we're waiting for people to answer questions, which I'm not sure if Eric is going, he may have accidentally pressed the button, but I will be in Australia starting on Monday. So if you're based in Australia, then please shoot me an email. I'd love to meet up with you if you do have any additional questions or you want to talk specifically about how your work might have use of Tapestri. I'm going to be attending the NDLR conference in Brisbane, and then we'll be hitting Sydney, Melbourne, and Adelaide. So if you are local to Australia and would like to chat further, please contact me. I posted my email into the chat box. It's ania.wronski@missionbio.com.
Thank you, Anya. Great. All right. It looks like we have all the questions today. If you have any additional question after the event, please don't hesitate to email us. The email address can be found from the chat box here, and I will go ahead to wrap things up. I want to thank all of our audience for taking time to join us today. And I certainly want to thank our speakers, Anya, Divya, and Dr. Tong, for taking their time and lending their expertise for such a wonderful webinar. So on behalf of Mission Bio and everyone there, thank you again and wish you all a great rest of the day.